In this video, we are going to study about pharmacodynamics. So, pharmacodynamics has been derived from two words, that is pharmacon and dynamics. Pharmacon means drug and dynamic means power. The power of the drug. So, pharmacodynamics is the study of drug effects. What drug effects are going to be produced in the body and how these drug actions or effects are going to be produced. That is pharmacodynamics. Now, there are certain basic principles of drug actions. The drugs, they do not impart new functions. They only alter the ongoing functions. They alter the functions of the body. They will only alter the pace of the ongoing function. Except in case of gene therapy. In gene therapy, you can also produce a new function, but otherwise the drugs, they only alter the function. The basic types of drug actions can be, first is stimulation. That is the ongoing function that is stimulated, the enhancement in the pace of the function. For example, adrenaline. Adrenaline will stimulate a specialized group of cells, that is the heart cells. It will stimulate the heart. So, stimulation is selective enhancement of the level of activity of specialized cells. Second basic drug action can be depression. Depression means decrease in the activity of specialized cells. For example, if adrenaline stimulates the heart, quinidine that depresses the heart. Or barbiturates, they have a depressant action on the central nervous system. Third is irritation. Irritation is a noxious action which can be produced in case of less specialized cells. For example, in epithelium or connective tissue. And if the action of the drug is strong and it has strong irritant action, that may cause inflammation or even necrosis of the area. The counter irritants or corrosives, they cause inflammation of the skin. Fourth is replacement. So certain drugs can be used as replacement of the natural hormones or metabolites in case of deficiency states. For example, in case of Parkinsonism, levodopa is given because our body is naturally deficient in levodopa. So in case of Parkinsonism, levodopa is administered exogenously. Or vitamins. Vitamins are exogenously administered in case of deficiency. Or iron. Iron is administered in case of iron deficiency anemia. Fifth type of action can be cytotoxic action. And this effect is basically on the invading parasites or the cancerous cells, which result in killing of these cells without affecting the host cells. For example, penicillin. Or cyclophosphamide. That has cytotoxic action on cancerous cells. Now we are going to discuss about different mechanism of actions. Mechanism of actions means that by what mechanisms or how the drug is going to produce its effects inside the body. Now, I have enlisted different mechanisms through which the drug produces its effect. Let's discuss the first one, that is the physical action. For example, isabgol. Isabgol is a drug. When we consume this drug, this goes inside the intestine, it absorbs water and swells to a larger size as compared to its original size. This is known as bulk forming capacity. So this is changing its physical character, it is changing its size, which is known as bulk forming property, and thus it is producing laxative action. So this drug is changing its physical character so as to produce the laxative action. Whereas antacids are the drugs which neutralize the gastric HCl. When we consume them, they are alkaline in nature. They go inside the stomach and they neutralize the gastric HCl via a chemical reaction. Thus, it has a chemical mechanism of action. Enzymes. Now, certain drugs have enzymatic mechanism of actions. Enzymes are basically proteins, but they are the prime target of drugs because most of the biological reactions which are going inside our body, they are carried out by enzymes. Enzymes are they have catalytic action and they are responsible for most of the biological reactions which are going inside our body. They can either stimulate or they can inhibit the rate of enzymatic reactions. Now, basically, to explain the mechanism, let us have one example. For example, this is an enzyme. Now, a substrate comes and attaches to this enzyme, and thus it produces a functional product or it produces a response. This is a stimulation process. In case of inhibition, there is an inhibitor which comes and binds to the binding site of the enzyme, and either it will produce a non-functional product or it will produce absolutely no product or no response. So this is inhibition of the enzyme. Enzyme inhibition can be of two types. The inhibition of the enzyme can be classified into two groups, that is non-selective and selective. Non-selective is, as I said, enzymes are basically proteins, so they can be denaturated by certain chemicals. And when they get denaturated, the functioning of the enzyme is totally inhibited, and these chemicals, they inhibit any kind of enzyme non-selectively. For example, strong acids or strong alkalis, they inhibit every kind of enzyme irrespective of any nature. Now, selective enzyme inhibition has two types, that is competitive enzyme inhibition and non-competitive enzyme inhibition. In competitive enzyme inhibition, as the name suggests, that there is a competition, and this competition is between the substrate and the inhibitor because they are structurally similar. The example which I earlier discussed, that this enzyme has a binding site and the substrate and the inhibitor have somewhat the same binding structure. If this is the substrate, it can go and bind here. Similarly, the enzyme can come and bind here. So compete for this single binding site. Whereas in non competitive enzyme inhibition, there are two sites. One is the binding site for the substrate, and there is a second site for the inhibitor, which is known as the allosteric site. So, if the substrate is binding here, but now the enzyme comes and binds here, it can inhibit the functioning of this enzyme irrespective of the binding site. So this is type of inhibition is known as non-competitive enzyme inhibition, because in this case the substrate and the inhibitor, they do not need to compete for the binding site. The examples of competitive enzyme inhibition are like dopa decarboxylase. It is an enzyme which has the substrate known as levodopa, and its competitive inhibitor is carbidopa. Whereas the example of non-competitive enzyme inhibition is aldehyde dehydrogenase. It is the enzyme which metabolizes alcohol, and the drug which is given to the patients of alcoholism, that is disulfiram, that is non-competitive inhibitor of aldehyde dehydrogenase. Now let us discuss about ion channels. As the name suggests, these are basically the channels, that is the transport system, which is allowing the passage of selective ions through them. As I told you, the membrane of the cell, and all the membranes in our body, they do not allow the passage of substances other than lipophilic substances. Non-lipophilic substances cannot cross through this membrane. So for the passage of these non-lipophilic substances, there is a presence of ion channels on them, and they allow the passage of selective ion through them. They remain in the closed state, and they can be activated. They have certain binding sites similar to the enzymes. They have binding sites, and if they bind with the stimulator molecule, they will open, and if they bind with the inhibitor molecule, they will remain in the closed state. For example, quinidine. Quinidine is a drug which inhibits the sodium channels in the myocardium. Now, transporters are similar to ion channels. They also help in the movement of ions and certain substrates, non-lipophilic substrates, across the membrane. But unlike that, ion channels, which were allowing the passage of the ions through them, these transporters, they carry these molecules, they bind to these molecules, and there is a conformational change, and they change their direction, and they throw these molecules inside the cell. And this movement can be according to the concentration gradient, or they can also pump the substrate or the ion against the concentration gradient using ATP molecules or metabolic energy. Next is receptors. Receptors, these are basically macromolecules, or these are binding sites, which are either located on the surface or inside the effector cell, and they help in the recognition of the signaling molecule or the drug. They help recognize the presence of the drug, and then they initiate a response. There are certain drug receptor interactions whenever the drug comes and it is recognized by the receptor, there are certain interactions between drug and the receptor. These are the drug receptor interactions. First among them is the agonist. Now, agonist is an agent which activates a receptor to produce an effect, and that effect is similar to that which is produced by the physiological signal molecule. Now, for example, this is a receptor, and this is the normal physiological molecule or the substrate, and it produces certain effect. For example, this substrate, it binds to the receptor, and the effect is rise in BP. Now, this agonist, it is somewhat similar in shape to the normal physiological substrate. It comes and binds to this receptor and produces the same biological response. So, this is known as the agonist. Now, the inverse agonist. The inverse agonist is an agent which activates a receptor, but it produces the response which is opposite to that of the agonist. For example, if the agonist was producing a response that was rise in blood pressure, now the inverse agonist, that will activate the receptor, but the response will be fall in blood pressure. Next is antagonist. Antagonist is an agent which prevents the action of agonist, and it will not allow the subsequent response, and it will not produce any response. The difference between inverse agonist and antagonist is the inverse agonist was producing the opposite response that to the agonist, but the antagonist, it prevents the response of agonist but does not produce any response of its own. Partial agonist, as the name is suggesting, it is agonist, but it is partial. It is not producing as much as a response as the agonist. For example, if the agonist was producing 100% response, now partial agonist will be producing submaximal response, and maybe it is producing 65% of the response or 60% of the response. So it is giving a submaximal effect. Ligand is basically, it is a molecule which attaches selectively to a particular receptor. It is very selective in nature, and it comes to bind to a particular receptor. It is basically, it is not any kind of interaction. It is just a term which is used to indicate the ability of a molecule to bind to a receptor. For example, the agonist and the competitive antagonist, they are ligands for same receptor. Now, there are different theories which explain the drug action through receptors, but the best explained theory is the receptor occupation theory. Now, this receptor occupation theory, which was given by Clark in 1937, it is based on the occupation of the receptors by the drug. That means more the number of receptors occupied by the drug, more will be the response. Now, basically, there is interaction between two molecules, that is the drug and the receptor. And this interaction is governed by law of mass action, and the effect, we see the effect is produced, is considered to be direct function of drug receptor complex. That means when these two molecules they interact, they will form a drug receptor complex, which will lead to the effect of the drug. Initially, it was designed as such, but subsequently, it was realized that just the occupation of the receptors, that means only the occupation of the binding sites on the receptor, is not sufficient. It is essential, but it is not sufficient to produce a response. What is actually needed is the agonist that is binding to the receptor that should be able to induce a conformational change. That means it should be able to activate the receptor. If we talk in scientific terms, there are two terms. One is affinity, one is efficacy. Now, affinity is the ability to bind. It is basically the binding ability with the receptor. Now, efficacy, which is also known as intrinsic activity, it is the capacity to induce a functional change. It will induce a functional change. So, affinity is the binding, which is essential. But does that agonist, they have the efficacy to produce a conformational change or to activate a receptor? Therefore, the theory was modified that the drug, it will bind to the receptor, and the drug receptor complex has to be formed. That means there should be affinity, but there is now a new term, that is strength of the stimulus, that is the efficacy or the intrinsic activity. Depending upon this strength, now the effect will be produced. At what pace, at what extent should be the effect, that depends upon the strength of this stimulus. Now these receptors, they have two type of essential functions. They have two types of functions. One is they are involved in the recognition of a specific ligand. They recognize that specific ligand and bind to that ligand. Second essential function is transduction. Transduction of a signal was binding of this ligand. Now transduction of that signal into a response, that means conversion of this signal into a response, that is transduction.